week, since it's the beginning of February, we start our Heart Month lecture series. We have 3 cardiovascular-focused series in a row, including February 13th on main campus in the Graham Center Ballrooms is our Heart Day event this year. So that's kind of an all-day event, which also includes our normal seminar. So hope you're gonna join us for that. And I can't think of a better person to help us kick off the Heart Month festivities, and it's not directly cardiovascular related, than our speaker today. So Dr. Gallego-Perez is coming to us from Ohio State University, where he is a professor in the Department of Biomedical Engineering and in general surgery, and he holds the Edgar C. Hendrickson Chair in Biomedical Engineering. He also serves as the director for the Advanced Nanotherapeutics within Ohio State's Gene Therapy Institute. So his research is all in kind of the nanomedicine space and the technology space, developing a lot of different platforms that cover a wide spectrum of physiology and diseases that you're going to see today. Really exciting research. And his research has been supported by a lot of different agencies and foundations, including NIH's Director's New Innovator Award, which is a very prestigious award that he got a few years ago. He started at Ohio State as faculty after he finished his Ph.D. there, and before that was a student in Colombia, which was where he's from. So he's also been a very big supporter of Latinx and BME, which has strong roots in our department, and has done fantastic work through their efforts, especially at the BMES. He serves as a fellow of the Biomedical Engineering Society, a fellow of AMPE, and at Ohio State, he received the Early Career Distinguished Scholar Award in Excellence for undergraduate research mentoring. So I know that student mentorship is extremely important to him. Very excited to have him here today to tell us all about his work.
Thank you for that introduction, Josh, and thank you everyone for showing up here so early. So again, my name is Daniel. I'm a professor of BME and surgery at Ohio State. And I'm going to try to tell you a few stories today about the work that we do in terms of rewriting cell fate. So I'll talk about cell reprogramming a lot. So for those of you who are not familiar with what my lab does, I don't run a very focused lab. We tend to be all over the place. Probably the only unifying theme is that we develop micro and nanoscale tools to be used in many different applications. We use them, for example, to study cell motility in the context of cancer and fibrosis. We also use them to develop gene therapies, for example, to treat cancer or metabolic disorders, or to develop cell therapies based on cell reprogramming to repair damaged nerve tissue both at the PNS and the CNS level. Today, my hope is to touch on 2 or 3 of these projects. I'm going to tell you a story where we use a gene therapy to repurpose tissues to potentially treat metabolic dysfunction. I'm going to try to make a connection between metabolic dysfunction and some brain disorders, specifically ischemic stroke and Alzheimer's disease. And then I'll tell you about a cell therapy that we have developed based on cell reprogramming to repair damaged brain tissue. And I'll touch briefly, very briefly, on some of the work we do at the PNS level as well. So this is the outline for my talk today. So as I said, I'm going to spend a fair amount of time talking about cell reprogramming. So I'll give you like a 30-second introduction to cell reprogramming. I'll tell you about direct reprogramming versus indirect reprogramming. I'll talk about some technologies that are used to drive cell reprogramming. These are mainly gene delivery approaches. And then I'll use that to introduce technologies that we have developed at OSU, which are cell and tissue nanotransfection, or TNT. And then I'll tell you about this concept of reprogramming tissues versus repurposing tissues, and I'll show you examples of how we use either one of those to drive therapies. And again, I'll focus on metabolic disorders, and then towards the end, if I have enough time, I'll tell you about how we can also use that to repair damaged brain tissue, both in ischemic stroke and AD. So cell reprogramming, for those of you who may not be familiar with the concept, is like alchemy, but with cells, right? This idea that you can take any cell type in your body, and for example, a skin cell, and then push that cell to become something else. For example, go from a fibroblast into a neuron, or fibroblast into a macrophage, or fibroblast into endothelial cells, right? So when you talk about cell reprogramming, most people probably have heard of the iPSC model, right? And that's where you take fibroblasts, you overexpress, let me just use my pointer here. Overexpress 4 transcription factors, OSKM, which are OCT4, SOX2, KLF4 and CMYC, and you can push that fibroblasts to become an induced pluripotent stem cell, which behaves a lot like an embryonic stem cell, but these are adult cells, adult tissue, right? And then once you have iPS cells, you can push those cells to become virtually any cell type in your body, right? Neurons, endothelial cells, you name it. Although we've learned a lot from this process, if you have worked with iPS cells, you probably know that this could be a labor-intensive process and also sometimes fairly inefficient process, right? So not every fibroblast that you transfect with OSPM will convert into an iPS cell. And that's not a major bottleneck right now because now iPS cells, we know that they can divide significantly, right? So you can, even if you only make a few iPS cells, you can expand them to large number to then be able to use them for different applications. The other issue is that once you have iPS cells, once you try to differentiate these cells into your target cell type, not every iPS cell listens to you and converts into your target cell type, right? Some of them do convert, some of them get stuck in between, some of them never convert at all, right? So having this mixed population of cells could potentially be an issue for many different types of therapies that people are trying to develop based on iPS cells. So because of those challenges, many labs, including our own lab, started to think of ways to bypass this iPSC station, and I forgot to mention, this iPSC approach is what we technically recognize as indirect cell reprogramming, right? You have to go to an iPS cell to then get to your target cell type. There's also something called direct cell reprogramming, right? And that's this idea that you can go straight from a fibroblast into a neuron, for example, bypassing the whole iPSC stage, right? So, for example, you can go take a fibroblast, overexpress these three transcription factors, ASCL1, BRN2, and MYT1L, and then push that fibroblast to convert into a neuron in a single step almost. You can also take a fibroblast, and we published on this a few years ago, overexpress ETV2, FOXY2, and FLI1, again, three transcription factors, and push them to become endothelial cells in a single step, right? So this is usually achieved by overexpressing different permutations of transcription factors, which are typically inspired by the developmental pathway of a specific cell type, right? So they change depending on the target cell type that you're looking for. Now there are many different ways to overexpress these transcription factors in cells, but most of the work so far on cell reprogramming has used heavily viral vectors for that. And viruses are great because they tend to be highly efficient, right? They have had billions of years of evolution to be able to perfect the process through which they inject nucleic acids into cells so that they can replicate, right? But when it comes to translational applications, viruses could still pose significant limitations. So because of those challenges, many labs have been working on developing non-viral approaches to gene delivery. There's some very elegant work on nanocarrier-based approaches using NMPs or polymer nanoparticles. My own lab also develops engineered extracellular vesicles for gene delivery, which I'm not going to talk about today. There's also some very cool work on the physical methods, right? So there's microinjection, gene gun, sonoporation, or electroporation. And within electroporation, I want to focus mainly on two technologies developed at OSU, right? Which are cell nanotransfection and tissue nanotransfection. In both cases, we use silicon nanochannels to shoot stuff into cells, essentially. So when I was postdocing at OSU, I spent a fair amount of time essentially taking cells from animals, bringing them to the lab, interfacing them with nanochannels that I fabricated in the clean room so that I can shoot stuff into those cells, and then putting them back into the mice to see how they responded to different conditions, right? As I became a faculty member, I thought, what can we do differently? How can we make this process more effective? So I thought, what about if instead of bringing the cells to the nanochannels, we find a way to bring the nanochannels to the cells, right? So we can do this process of gene delivery in vivo without perturbing the natural microenvironment. And that's when we came up with the concept of tissue nanotransfection, or TNT. So this is us a few years ago, pretending that we are tissue nanotransfecting a human. This is my very first research assistant, Alec. He has a TNT chip on his forearm. It's a silicon chip. If you were to zoom in into that location, what we essentially have is millions of skin cells, because the chip is sitting on the skin surface, of course, directly interfaced with silicon nanochannels, roughly at a 1-to-1 ratio. When you apply a potential, and then those nanochannels, of course, can be loaded with any cargo of interest. In this case, let's think about reprogramming genes as an example. When you apply a potential across that system, what we essentially do is we create an opening on the cell membrane, we porate the cell membrane, the size of the nanochannel, right? So it's a tiny hole in the cell membrane. And then because this DNA has a negative charge, you can set up the polarity of the electrodes but you can push that cargo into the cell by electrophoresis, right? So essentially that DNA gets shot into the cell through an active process of electrophoretic motility, which looks more or less like this is an in vitro example. Up here we have labeled DNA, down here we have a cell that's not visible right now, and in between we have a nanochannel, right? And when we apply a potential, again, we create an opening on the cell membrane, and then that cargo translocates from top to bottom into the cell, right? So it's a fairly effective process to deliver cargo into the cell, and that's also very benign. One huge differentiating factor between nanochannel-based poration versus a standard electroporation is that in electroporation, typically the entire cell membrane, as you can see in this simulation, gets exposed to a high-intensity electric field, right? Which causes a kind of widespread pore formation on the cell membrane, which could be very detrimental for the cell. When you interpose a nanochannel between the electric field source and this cell, you essentially end up focusing that electric field on a tiny portion of the cell membrane, right? As you can see in this case, that portion is only this area right here, which is about less than 2% of the surface area of the cell, which makes this technology very benign. And then, of course, because the channel has high electrical resistance, you maximize the voltage, which means that the transmembrane potential in nanochannel-based poration tends to be higher compared to bulk electroporation. So when you talk about electroporation, when you achieve high transmembrane potentials and when you also focus them on a small portion of the cell membrane, you tend to achieve cell viability and transfection efficiencies that are close to 100%. Now going back to the use of this for reprogramming purposes, right? So if we use TNT to deliver reprogramming genes into skin tissue, for example, what we essentially do is we turn the skin into a bioreactor that can grow other tissues, for example, neurons, endothelial cells, adipocytes, you name it, for downstream applications. Now, a few things that are important to highlight here is the fact that first, we use nanochannels to deliver the genes into the tissues. We don't have to use viruses. And this procedure takes only about 100 milliseconds, right? So we only touch the skin once and we retrieve the device and we're done, right? And that's gonna be important information for later. So these are a couple of examples where we use TNT to deliver reprogramming genes into skin tissue to drive in vivo cell reprogramming. So as I mentioned at the beginning, if you take these three transcription factors, ASCL1, BRN2, and MYT1L or ABM, which have been shown to convert fibroblasts into neurons, we can do the same type of conversions, but in this case in vivo. The first thing that we did though was to compare or to benchmark TNT to standard bulk electroporation, right? So this is a graph showing gene expression. The y-axis shows expression levels as relative to bulk electroporation. And then you have the three genes, and as you can see, for all three genes, we get anywhere between 50 to 250-fold higher expression levels compared to standard bulk electroporation. These are a couple of histologies of the skin after TNT, a few weeks after TNT. The green color is essentially a marker for the skin cells, K14, and the red color, TUJ1, is a marker for neurons. And in the control skin, which was TNT with empty plasmids, we do see some red signal, and that's because, of course, the skin has nerve fibers, right? So you're expecting to see some TUJ1 signal in the skin. But when you do transfection with ABM, the cocktail that makes neurons, you see that the amount of red increases significantly. You see a lot of accumulation around the hair follicles. We did lineage tracing experiments, and we found that these neurons that develop in the skin, they are derived from the keratinocytes in the epidermis and also from the fibroblasts in the dermis. And then we also did some action potential measurements, and we found that those neurons actually fire action potentials, which basically indicated to us that they were able to mature within the skin, right? And you're not supposed to have neurons in the skin. So we were hesitant to pursue this because we thought those neurons were going to die eventually, but no, they were able to be induced and also mature. Down here, there's another example of cell reprogramming happening in the skin, but in this case, we use a different cocktail. So as I mentioned, we found that when you overexpress ETV2, FOXY2, and FLI1, or EFF, you can make endothelial cells from fibroblasts. So when you use TNT to deliver those three genes into the skin, this is some histology of the skin. Control skin again was TNT with sham plasmids, and this red PCAM1 and GWF2 endothelial cell markers, right? So you do see some red signal and green signal in your control skin, again, because skin tissue has blood vessels, so you are expecting to see some of that as well. But when you do the EFF tissue nanotransfection, you see a lot more red and a lot more green, indicating that there's new endothelial cells being generated within the skin. We also did lineage tracing experiments to try to identify where they came from, and we found that only fibroblasts contribute to endothelial cell formation through cell reprogramming. Keratinocytes, no matter how much you hit them with the EFF cocktail, they never convert into endothelial cells. And we can discuss that later if you are interested. Now, we knew we can make endothelial cells in the skin, right? But can they assemble into blood vessels? Can they be used to potentially perfuse areas that are hypoperfused? So we tested that hypothesis in a fairly aggressive model of tissue ischemia in mice. So we use something called hind limb ischemia, where you essentially take a mouse and you remove a significant portion of the femoral artery of that mouse. So when you do that, of course, if you do laser Doppler imaging of the limb to look at perfusion, you look at the limb and it looks very hypoperfused, as you can see in this picture over here. And then, of course, if you don't treat that limb is going to end up necrotizing. As you can see in this case, most of those mice tend to lose their limbs because of the heavy ischemic insult that they receive. But when you do the same, when you do induced ischemia, and then you stitch the skin back together, and then you apply TNT on the skin surface to make new blood vessels, we are able to create new collaterals, as you can see from this laser Doppler image, that originate in the skin and then end up reperfusing the entire limb, right? So you can see this limb is very perfused compared to the control case. And then, of course, those limbs that we treat with TNT, they do not necrotize, they don't die, right? There's some necrosis happening, as you can see here toward the tip of the toe, but overall that limb looks really healthy compared to an untreated limb, right? So this shows us that we can indeed not only make endothelial cells, but that these endothelial cells can assemble into blood vessels and reperfuse heavily ischemic tissues. Now, I like cell reprogramming a lot, right? I think it's very interesting from a scientific perspective, but I also spend a ton of time thinking about translation in my lab. I want to make sure that we develop things that could eventually help patients, right? And although I still firmly believe that cell reprogramming could be a solution for cell and tissue scarcity in many different types of applications, I have to recognize that there's still many barriers that we have to overcome for cell reprogramming to make it to the clinic, right? One of them is that this is a very stressful process, right? I cannot tell you how many times I spent as a postdoc doing time-lapse imaging, trying to see how a fibroblast converts into a neuron to see the whole thing fail, right? So you start to see the fibroblast sort of condense the cell body, and you get excited, right? Because that's usually an indication that successful conversions are happening. You start to see the fibroblast extending processes from the cell soma, you also get more excited. But then the cell decides to undergo apoptosis, right? Many of these cells rather kill themselves than to commit to a different phenotype. And we haven't figured that out yet, but it happens very frequently. Also, and I get this question asked a lot when I talk about cell reprogramming, we don't know how stable these phenotypes are over time, right? I haven't seen so far a cell revert back to a fibroblast, right? So once you commit that cell to an endothelial cell or neuron, I've never seen it go back to fibroblast, but never say never, right? You don't know. You have to de-risk this heavily before you can actually use this in the clinic. So because of those challenges, and again, because my lab wants to think of translation all the time, I started to consider the idea that maybe we shouldn't be doing reprogramming in some cases, maybe repurposing tissues is enough. And what do I mean by that? So in tissue repurposing, you don't really have to change the identity of the tissue, right? The whole idea is to essentially channel the function of one tissue through the skin, for example. So for example, force skin tissue to behave like adipose tissue, but skin tissue is still skin tissue, right? The identity doesn't change, the function doesn't change, right? That may have less barriers to clinical translation. So I'm gonna tell you a story where we use this concept of tissue repurposing through a gene therapy to potentially develop therapies for obesity and metabolic dysfunction. So I don't have to spend too much time introducing this to this crowd. You guys are very well informed, of course, of type 2 diabetes, obesity, metabolic dysfunction are growing rapidly worldwide, right? They are a huge burden to the healthcare system. There's a multi-million-dollar industry right now, multi-billion-dollar industry actually, focused on trying to find solutions for this epidemic. There's a lot of work and a lot of promise, for example, in GLP-1 agonists, but there's also a lot of drawbacks that people don't talk about with GLP-1 agonists. One thing that the field has known for a while, though, is that the solution to this epidemic could be within our own bodies, within our own tissues, right? And what do I mean by that? So, if you don't know your facts, essentially we have two different types of adipose tissue. We have white adipose tissue, which fulfills a physiological function, but excessive amounts of white adipose tissue can be detrimental to metabolic health, right? We also have something called brown adipose tissue, or BAD. in excessive amount of spread adipose tissue tends to correlate with improved metabolic health, right? And that is in part because brown adipose tissue has something called thermogenic activity, which means it drives energy expenditure through thermogenesis, and then it also has some secretory roles or endocrine-like activity, which I'll come back to later. So there's promise in brown adipose tissue. The challenge, though, is that we as adult humans, we don't have enough of it, right? To be able to use it to try to treat metabolic dysfunction, right? And every attempt that has been made to increase the mass of brown adipose tissue or the activity levels has failed significantly. Right, so this to me looked very much like a reprogramming problem, right? You don't have enough brown adipose tissue, so let's make some out of skin tissue. So I'll show you one example of that, but that's not what I really want to focus on in terms of tissue repurposing. So there have been some reports that these two genes, PRDM16 and CBP beta, when you overexpress them in myoblasts, you can induce the conversion from a myoblast into a brown adipocyte, right? So we tried to use TNT to deliver those two genes into the skin of mice, and then we looked at reprogramming to our brown adipose tissue, and this is work that was initially led by former Ph.D. student in my lab, Devlina, and currently led by Ph.D. student Natalia in my lab. So when we did histology of the skin, UCP1 is stained in brown in this case. You can see that the skin that was TNT treated with these two genes has UCP1 expression around the hair follicles, which are absent in the control skin. So this gave us a positive indication that maybe we are making brown adipose tissue from skin tissue, in this case, to some reprogramming. We did PET-CT scan to determine the biodistribution of that adipose tissue. And as you can see, this area that's surrounded by these red arrows, that's where we did the TNT on dorsal skin. And that's the only area where we see significant development of brown adipose tissue, right? So we saw that this was very confined to the area that we had TNT treated. I also mentioned that this tissue is supposed to have thermogenic activity. So the way we tested that is we put a cold stimulation on the skin of these mice, and then we measured the amount of time it took for that skin to go back to body temperature, right? Because bat tissue responds to cold stimulation through thermogenesis, right? So it generates heat. So we have this graph over here. In the y-axis, we have the amount of time it took the skin to go back to body temperature. And in the x-axis, we have the amount of time it has passed after the TNT intervention. So 7 days, 14 days, 21 days, all the way up to 28 days post-TNT. So when we look at the control skin, this was TNT with sham plasmids or empty plasmids. Regardless of the time post-TNT, you can see that the amount of time it takes to go back to body temperature is about 4 minutes, right? So at day 7, all the way up to day 28, we didn't see much change. It took about 4 minutes for this skin to go back to body temperature after the cold stimulation. When we overexpress these two genes, where we can actually make UCP1, as you can see, as time goes on, the amount of time it takes for that skin to recover decreases significantly, right? So at day 28, it takes only about a minute for the skin tissue to go back to body temperature, right? Which indicates that we are indeed driving thermogenesis locally in these skin patches. And then recently, we also made a modification to this reprogramming cocktail. We added one more factor, and we have done some glucose tolerant tests. And as you can see, the TNT, the tissue that was TNT with these 3 factors now has also driven improved glucose tolerance, right? But again, that's not what I wanted to focus on when I talk about tissue repurposing. So let me just refocus my talk. I presented that data that I just showed you at a seminar at Ohio State, and then Kristen, which is a collaborator of mine, approached me, and she's an expert in brown adipose tissue, and essentially she's been working on that other function of brown adipose tissue. As I told you, it has thermogenic activity, but it also has endocrine-like activity, right? And Kristen is an expert in the endocrine-like activity, right? Turns out brown adipose tissue makes metabolites, which are typically small lipids, which are called lipokines or barokines, and those metabolites can signal many different types of organs and tissues to drive positive effects, right? One type of lipokine is oxylipids, right? And within oxylipids, we have 12-13-dihone, which is a lipokine that Kristen has spent her whole career trying to study. So Kristen knew that when you exercise or when you are exposed to cold stimulation, 12-13-dihone increases typically in circulation. This is based on mouse data. And that has a positive effect in liver health, cardiac health, and many different aspects, right? The challenge they face is that when they injected that lipokine directly into the circulation of these mice, that lipokine got metabolized very quickly, right? Within minutes, it was gone from circulation, right? So it never had enough time for it to actually drive a positive effect in those mice. So Christian said, what if we use your technology, TNT, to deliver the lipokine into the skin, right? To see if we can actually be more successful trying to upregulate the lipokine. And I told Christian that that's gonna fail, right? I told her if you fail at upregulating the lipokine by directly injecting that into the blood, I don't think we're gonna be successful just by delivering a few thousand copies of the molecule through TNT into the skin. I told her, what if we force the skin to make the lipokine, right? So what if we use the skin, we repurpose the skin to make it function like brown adipose tissue, at least partially. So I asked Christine, what are the most important genes in the biogenesis of the lipokine? And she pointed to 4 enzymes, EPHX1 through 4, and out of those, EPHX1 and 2 are the most important ones. So essentially the way this works, linoleic acid enters the cell, and once linoleic acid, which you can consume in the diet, enters the cell, the brown adipocyte, it gets metabolized or processed by cytochrome P450 into a metabolite called 1213 EPOM, and then EPHX1 and 2 enzymes essentially hydrolyze that metabolite to make 1213 dihome, which is down here, and then 1213 dihome can exit the cell, and then again signal many different types of tissues and drive all these positive effects that they are trying to emulate. So essentially what we set up to do, and one thing to highlight is that in this process, when you exercise or when you are exposed to cold stimulation, this portion over here gets boosted, and that's what produces an increase in the lipokine production when you get exposed to that. So our goal here was fairly simple, right? What if we use TNT to overexpress these 2 genes in the skin? The skin has already all the other components. The skin has linoleic acid, and the skin also has cytochrome P450. So we were just providing the skin with the 2 enzymes that it was missing to see if the skin can make the lipokine itself, right? So again, we're not changing the identity of the skin. We're just tricking the skin into behaving like brown adipose tissue, at least for some time. So the way we tested that hypothesis, essentially, we took some mice and we exposed them to a high-fat diet for 6 weeks. And then between week 6 and week 12, we did TNT on the skin surface once a week for 6 weeks, essentially, right? And then we looked at multiple parameters to see if we were enhancing metabolic health. The first indication that we were onto something is when we measured the lipokine levels in circulation, right? One thing that I didn't mention is that we did do TNT for 6 weeks, so up to week 12, but then we waited for 2 weeks to measure the lipokine in circulation, right? So these measurements of the lipokine in blood were taken at week 14. And as you can see, the group that was TNT treated with the EPHX1 and 2 plasmids shows a significant increase in the lipokine in circulation, which is not present when we TNT with sham plasmids or when we TNT with UCP1 plasmids, right? So this was a first indication that we are indeed successful at forcing the skin to make the lipokine. We saw that those mice that were TNT treated with the EPHX1 and 2 plasmids, they had a tendency to gain significantly less weight over time, right? So these are these mice down here. They do gain weight, but less compared to mice treated with UCP1 plasmids or mice treated with sham plasmids. Those mice also tend to have decreased fat mass and increased lean mass. And when we looked at liver tissue health, we saw that the mice that were treated with sham plasmids, they tend to develop fatty liver disease, as you can see in these pictures down here. The mice that were treated with the EPHX1 and 2 plasmids, they tend to maintain a fairly healthy liver, as you can see in these pictures up here. And we also looked at systolic function and diastolic function, and both of those were also significantly improved in response to this therapy, right? So I know I told you that we had to do this treatment for a total of 6 weeks, right? Once a week for 6 weeks, which may sound like a lot, right? But think about this. What if I told you for the next 12 weeks, you can eat anything you want to eat, a high-fat diet if you want to. And then between week 6 and 12, you only have to work out once a week for 100 milliseconds. And then that's enough for you to maintain healthier weight, healthier liver, and improved cardiac function, right? It doesn't sound like such a burden, right? So we are essentially trying to reap the benefits of exercise without breaking a sweat, right? So we expanded the potential use of this approach to other conditions. Turns out brown adipose tissue also has a lot of beneficial effects in aging. So we recently published a paper where we did the same type of procedure, but in this case, we didn't do it on high-fat diet fed mice. We just did this in old mice, so these were 21-month-old mice, and we did again TNT for 6 weeks. And then we looked at the lipokine levels in circulation, and we were able to upregulate the lipokine in the old mice circulation, and that of course led to a bunch of beneficial effects in terms of cardiac health, right? So when we looked at ejection fraction, ventricular wall thickness, hypertrophy index, and diastolic and systolic function, all of those were significantly improved in response to this therapy. So again, think about how much damage you tend to get from aging, right? So 21-month-old mice are fairly old mice, and only 6 weeks worth of TNT treatments was able to revert back a lot of the damage caused by aging in the hearts of these mice. So just another application to highlight. How am I doing on time? So these are some conclusions up until this point. So TNT can be used to derive lipogenic function from the skin, and that function can lead to improved cardiometabolic health and acting very much like an exercise patch, right? So forcing the skin to derive the benefits of exercise without actually having to work out. TNT is localized on the skin, but it could have a systemic effect depending on what we overexpress in the skin. And then, as I mentioned, there's potential additional applications. One of them is mitigating the effects of age-induced cardiac dysfunction. And we have done some recent studies, still very preliminary, that indicate that it can also be used to improve maternal, fetal, and offspring health. But that's more to come in the future, right? We're still very early in that studies. Now, as I mentioned, metabolic dysfunction is a huge burden to the healthcare system. That's in part because it could be a precursor for many other types of conditions, right? So complications in pregnancy, eye complications, nerve damage, kidney disease. Of course, many people are familiar with cardiovascular disease. And within that category, we also have increased risk for reduced cerebrovascular function, right? And of course, when you think about reduced cerebrovascular function, most people tend to think of increased propensity for ischemic strokes, which is a huge factor, of course. But there's also emerging evidence suggesting that reduced cerebrovascular function can also be connected to Alzheimer's disease or related types of dementia, right? So I'm gonna use the last maybe 20-25 minutes of my talk to tell you about 2 studies where we have used cell reprogramming in this case to try to address some of these conditions, specifically ischemic stroke and Alzheimer's disease. So I'm gonna come back to this example where we use TNT to overexpress discrete transcription factors to make endothelial cells, and I show you how we can use that to counteract ischemia and necrosis in this model of hindlimb ischemia. But we wanted to see if this approach can also be used to potentially drive repair of damaged brain tissue, right? And it's not a stretch, right? I'll show you an example of what we've done in the PNS to try to make the case for how this could work in the CNS. Of course, when you make new blood vessels and you essentially do that in ischemic tissue, you get the benefit of reperfusing the tissue, right? You bring blood flow again back into the tissue, that's good. But when you also make blood vessels, there could be other benefits that could be used to repair or regenerate other types of damaged tissue. And one of those tissues is nerve tissue, right? And the reason that's important is because during development and also in response to injury, people have found that blood vessel formation is highly synergistic with nerve tissue repair and regeneration, right? So when you create new blood vessels, those blood vessels typically act as a scaffolding structure for neuronal cells to migrate along or to extend axons along. And these endothelial cells are also known to release neurotrophic factors that again play a critical role during tissue development and also in response to injury, right? So when you make blood vessels, you indirectly also foment nerve tissue repair and regeneration. And I'll show you an example, a fairly simple example, but I think a very effective one to convey this message, right? So we took some mice and we applied a crush injury in the sciatic nerve, right? So we crushed that nerve, and then we used the TNT technology to deliver the same cocktail that makes blood vessels, the EFF cocktail, hoping to make blood vessels in the crushed nerve. And then this is some histology of that nerve. So we have the control tissue on top, which was TNT with sham plasmids. Over here in red we have vascular tissue, and in green we have axon staining, neurofilament in this case. And as you can see in the control case, we don't have any vascular tissue, of course, and the axons are able to grow back across that crush site, but they do so in a highly disorganized manner, right? When we use TNT to make new blood vessels in the crushed nerve, we are able to grow blood vessels, you can see in this picture, but they do so in an aligned manner, right? And then the axons essentially follow that same pattern, right, of aligned repair and regeneration, which then leads to improved muscle function. This was again tested in a fairly simple model of crush injury. It was led by former Ph.D. student in my lab, Jordan, in collaboration with Dave Arnold. But we recently, under DoD funding, we extended this to much more complex modes of nerve damage. In this case, in collaboration with Amy Moore, chair of plastic surgery, and work led by my postdoc Anna and a few of my students, Sarah, Megumi, Alejandro, and John. We were able to use this approach of making blood vessels to enhance nerve repair and regeneration in segmental nerve defects, right? So in this case, we don't crush the nerve, we remove a chunk of the nerve, and then we have to repair that with a graft, and then we apply TNT on the nerve to generate blood vessels. And as you can see in these quantifications, that leads to improved muscle function and also nerve function. So again, this is just an example trying to make the case for how we can use blood vessel formation to foment the repair of damaged nerve tissue. But I'm gonna try to show you that this can also be used to repair damaged brain tissue. So coming back to the brain, right? So as I mentioned, obesity, type 2 diabetes can lead to many different types of conditions. This could increase the propensity for ischemic strokes. Most strokes tend to be ischemic in nature. And ischemic strokes essentially is where you have a blood clot that blocks blood flow to your brain. And then that, of course, the ischemic insult causes a lot of cell deficits, which then lead to a bunch of issues like paralysis, visual and speech impairment, and death in about 42% of the cases. The problem here is that there's really not a ton of therapies options for ischemic stroke, right? The only approach is essentially a thrombolytic approach, which essentially means getting rid of that blood clot mechanically or enzymatically. And if the patient shows up to the hospital within the first 6 hours, they tend to use chemical methods to try to dissolve that blood clot. Again, the goal is to try to reperfuse the brain as quickly as possible to mitigate the damage, but there's some damage that already occurs, right? If you show up to the hospital within the first 24 hours, you might still be eligible for this type of therapy, right? But they might have to resort to mechanical methods to disrupt that blood clot. And that mitigates or contains the damage in the brain, but there's still some damage in the brain. If it's been more than 24 hours, you're essentially out of luck, right? You're not longer eligible for this thrombolytic therapy. So there's already a lot of damage that has occurred in the brain. And of course, the bigger the damage, the higher the chances of death. But regardless, for all 3 cases, there's damage that already occurred in the brain, small or large. And there's really no therapy to revert that damage back, right? So that's where we're trying to make a difference. So this is work that was done in collaboration with Shai Nimji, a neurosurgeon at Ohio State, Dave Arnold, again, a neurologist, and then Natalia, who also happens to be my wife, she's also a professor at Ohio State. And we had a fairly simple but ambitious idea, right? And we thought, what if a patient presents to the hospital after the 24 hours, right? So when they no longer are eligible for any type of thrombolytic therapy, what if we can take a skin cell from that patient? What if we can prime those cells to become endothelial cells and somehow deploy them back into the brains of those patients to see if we can foment blood vessel formation and somehow also foment the repair of damaged brain tissue, right? Just based on the evidence that we had collected in the PNS. So this work was led by former PhD student in my lab, Luke Lemmerman. And it's a fairly long story, but I'm gonna try to make it very short here to give time for questions. So Luke took some mice and he induced ischemic stroke in those mice using the MCAO method. And MCAO essentially stands for middle cerebral artery occlusion, right? So you occlude a blood vessel and you induce a huge, massive stroke, typically in the right hemisphere of the brain of these mice. Then 6 days post-stroke induction, we took skin cells and we used nanochannels to inject the EFF Coulter into those cells. And then 1 day post-transfection, we injected the cells into the brains of those mice, right? So right above the cortex of the stroke-affected hemisphere. So 7 days post-stroke, so a lot of damage has already occurred within this time window, right? So we wanted to recreate an environment where we essentially had already created a ton of brain damage, but we were also waiting for the inflammation to go down before we actually deploy our cells into the brain. So then we looked at stroke size, 14 days post-stroke induction, so essentially 7 days post-LV injection, and you can see that the stroke size has reduced a little bit compared to what you have at day 2, but the stroke is still fairly large for the mice treated with control cells. The stroke is a lot smaller for the mice treated with EFF transfected cells. When we look at histologies of the brain, and we have the controlled brains up here and the EFF treated brains down here. When we looked at neuronal markers in the motor cortex, you can see that the motor cortex of these mice have a lot of cells, but the neuronal markers only show up in this portion, right? So in red you have neurons here, but you have a huge chunk of the cortex which doesn't have any neurons, right? It has cells for sure because you have DAPI positive elements in there, which means that there are nuclei of cells in there, but there's no NeuN expression, so meaning no mature neurons are present in this portion of the cortex. When we look at the brains of the mice that were treated with EFF transfected cells, though, the motor cortex of those brains, they are full of neurons, as you can see in this picture, right? When we looked at scar tissue formation based on GFAP staining, we see a lot of scarring in the brains of mice treated with control cells and less scar tissue formation in the brains of mice treated with EFF transfected cells. Of course, I can show you a bunch of 3D histologies, but if we don't make a difference in terms of behavior, we are not really making an impact in this study. So, of course, as I mentioned, when you induce a stroke, a huge part of the brain that gets impacted is the motor cortex, right? So these mice have a ton of motility impairments. And so the way we measure that is through many different tests. I'm just going to show you results from an open field test where you essentially put the mice in a box, then you trace their motion, and then you can use a bunch of different tools to try to calculate whether they have recovered some of that function. So this is a couple of videos of mice treated with the control cells versus EFF transfected cells. On that side, we have a mouse treated with control cells, and I'm going to play that video. So that mouse can barely move, right? Probably has a massive headache still. This was 21 days post-stroke induction, right? On the right side, we have video of a mouse that was treated with EFF transfected cells, right? So again, the mouse seems to move a lot easier compared to the control mouse. Of course, we can quantify a bunch of parameters from those videos, and I'm going to show you those quantifications now. Virtually by every metric that we looked at, all these mice that we treated with the EFF transfected cells show significant signs of recovery, right? In many cases, to levels pre-stroke induction, right? So almost as if they never had a stroke. Right, so this was very promising to us because, of course, we were able to show we can repair damaged brain tissue, as you saw in those pictures where we had more neurons present in the case of the EFF treated mice. But on top of that, we also got significant recovery in motor function, right, which is not easy to achieve when you have a massive stroke that is not treated. I'm going to use the last 5 minutes maybe to tell you one last story, maybe half of the story. As I mentioned, there's emerging evidence suggesting that there's reduced cerebrovascular function can also be linked to Alzheimer's disease, right, and other types of dementia. So I have a postdoc in my lab who has a neuroscience background, Diego, and he wanted to test whether these benefits that we saw in the stroke model could be recreated to some extent in a model of Alzheimer's disease, right, of dementia. And again, the rationale was fairly simple, right? Can we make blood vessels in the brain to somehow foment the repair of damaged brain tissue to try to regain some function, right? And in this case, it will be cognitive function, right? Memory is what we look for when we think about Alzheimer's disease. So this was just recently published in a preprint. I'm going to direct your attention to that if you want to read the whole preprint. I'm just going to show you one set of results, mainly behavioral data, but there's a lot of data we have collected to show how these cells operate in the brains of those mice and how they potentially drive recovery. So we did one experiment where we went in very aggressively, right? So we had mice that had Alzheimer's disease. We used a model called the Ph.D, which has 3 genetic mutations that drive this condition in mice. And we injected cells into the ventricles of the brain of those mice 3 times, right? Spaced by 4 weeks each injection, right? So we went very aggressive because we didn't know we were going to have an impact in terms of cognitive function. This is work done in collaboration with Nikki at Ohio State. So we did many different tests. I'm going to show you the results of the Barnes-Maze test. If you're not familiar with that test, the way it works is you essentially put mice on a platform which has a bunch of openings, but only one of them is an escape hole, right? So when you scare the mice on that platform, that mouse essentially is gonna try to hide, but of course it only has one hole where it can hide. So the mice spend some time trying to learn this task. They tend to learn where the escape hole is, and when you repeat that multiple times, they will essentially, when you scare them, they will go straight into the escape hole. So you then retrieve the mice from that platform, then you put them back on it a few days later. If the mice don't have cognitive deficits, they are supposed to remember where the escape hole is, right? So when you scare them again, they will run back to the escape hole. If they have cognitive deficits, if, for example, mice with AD, they will forget, and they will have to relearn the task again where the escape hole was, but they can actually hide when you scare them. I'm going to show you two videos of mice with Alzheimer's disease. In one case, we have a mouse treated with controlled cells. In the other case, we have a mouse treated with EFF transfected cells. The escape hole on that platform is essentially here. These dashed lines show where the escape hole actually is. So let me just play the video of the control mouse, right? So again, this is a mouse with AD that we treated with control cells. And you can see the mouse starts to probe the entire platform again, right? Because that mouse forgot where the escape hole was, right? So eventually it will make its way to the escape hole and stay there, essentially, right? Because that's where they want to go once you scare them. On this side, we have a mouse that was treated with EFF transfected cells, and you can see that mouse will just rapidly run to the escape hole, right? It tends to remember where the escape hole was, which gave us an indication that this therapy was potentially improving the spatial memory in these mice. Of course, I can pick any video to fit my narrative, so I want to show you, of course, the actual quantitative data. And I want to direct your attention to healthy mice first. So, of course, we did this test in both mice that had the disease and mice that didn't have the disease, right? So, wild-type mice, the ODT in this case, when you inject them with these EFF transfected cells, you don't have an effect, right? You don't give them superpowers or super memory, right? So if they don't have any deficit, they tend to remember where the escape hole was, regardless of the treatment that you do on those mice. Mice that have AD, which will be the 3xTG AD mice, as you can see, those mice tend to travel a smaller distance to the escape hole, right? So they tend to go to the escape hole directly compared to mice treated with control cells. Those mice also tend to make less errors in trying to find that escape hole compared to mice treated with control cells. And those mice also tend to spend more time around the escape hole compared to mice with control cells, right? So again, this gave us good indication that this therapy that we had developed initially to treat damaged nerve tissue in the PNS could also repair damage after a stroke and also repair damage in models of Alzheimer's disease. So this is the last slide I have of conclusion. So in this case, vasculogenesis driven by nanotransfection can promote and guide nerve tissue repair and regeneration both at the PNS and CNS level. And of course, this can lead to increased perfusion, which improves muscle function and motility, and also memory retention depending on the type of model that you're testing this in. So I'm going to use the last couple seconds to make sure that I acknowledge the work that everyone did to get us to where we are at. So we have current lab members, alumni from the lab on the right side, all the collaborators of mine for the project that I presented today, and all the funding that has supported the lab for the past 10 years. And of course, thank you for your attention. I'll be happy to take any questions.
I have a question. First of all, thank you for sharing all of that. I think as biomedical engineers, we always aim to develop methods that can be applied to a variety of things. I think you've done just that, so thank you for sharing that. I have a very philosophical question, which I'm going to probably have a hard time formulating, so please ask me for clarification if need be. And it has to do with the theory of reprogramming versus repurposing, right? A lot of times, we define a cell type based on its function, right? So if I take a fibroblast and I make it express TUJ1, is it a neuron? Does it have to have these dendritic processes? And then the visual cell is defined as, cells lining the inner layer of if there's a cell expressing again TUJ1 in the visual layer, is that in the visual cell? So, and how do you think about that philosophy? And then in terms of reprogramming versus repurposing, if you're getting skin cells to act as adipose tissue making this brown fat, how is that different from reprogramming?
So, to the first point, the way we typically do that is we define it based on function, right? So when we look at reprogramming studies, we of course look first at markers, right? That will define a cell identity, but markers on their own are not enough for us to make sure that this is a successful conversion, right? So when we make neurons, we always do patch clamping to make sure that they can fire action potential, right? Because it's not enough to just have neuronal markers being expressed in those cells and to even have the morphology, right? You have to have the function, right? So that's why even when we did the endothelial cell reprogramming, it wasn't enough for us to just see a bunch of markers of endothelial cells in the skin. We wanted to see that there was actually also ability to create blood vessels that can reperfuse, right? So we always think of function from that perspective. And sometimes partial function can be enough for what we're trying to achieve, right? But function is usually what defines to us whether we were successful or not in converting one cell into a different cell type. For the other question, yes, I mean, I think it depends on what you want to achieve, right? So what we've done so far is we can force skin tissue to recapitulate one aspect of renal tissue, which of course seems to have a ton of benefits, right? But renal tissue is a very complex tissue, right? It has many different types of functions, right? So you always ask, what if we are able to recapitulate all of the functions, right? So you also have to balance what's feasible and what's easy to translate and what isn't, right? So that's why it's good that you are able to recapitulate maybe one function and cover a ton of bases, but it's not the entire identity of the tissue, right? So there's always that question, what if we are able to do an extra function, or we are able to recapitulate a function, right? Are we gonna be more successful or not? And maybe the answer is not. And then that's a good exit for you to say, well, this is enough. But until you actually run the studies, you don't really know if you need to recapitulate all functions to be able to drive more effective therapies. Does that make sense? And maybe just one thing to highlight is when you do reprogramming, as I mentioned, those changes tend to be permanent. You have to de-risk it, but they tend to be permanent, right? One intervention can make pre-adipose tissue. If we only do repurposing, we have to reintervene, right? Because it's just temporary, right? We have to give the cells again the genes that they can express, so they can make the lipokine, right? If we don't continue to do that, eventually it will stop functioning, right? So when you do reprogramming, there is only one intervention, and hopefully it will last forever. So there's pros and cons of going with either one of them.
Thank you. Is there some benefit to the repurposing in that if things go badly, you might also be able to either wait it out or even shut it off if you thought about that?
Yeah. So part of the benefit of not using viral vectors is that this is supposed to be a transient expression. So if something goes wrong, I mean, you just wait it out. Also, we are very intentional in terms of using the skin as a conduit to drive this function, because skin can be monitored and skin can be excised if you need to. So if something goes wrong, if you see behavior that is problematic, then you can just excise that patch of skin tissue, and then your problem is technically done, right? It will be different. We have to do this in the liver or something that's very difficult to access. So yeah, so it has benefits from that perspective as well. Good question. Yeah.
So, and the little I know about like gene regulation is there's a lot of different steps and things that happen with upregulating and downregulating a gene inside of a cell. So with you're basically throwing the gene, the DNA, into the cell, and the cell is incorporating that into its own genome, or it's just using the machinery from the DNA polymerase and everything to just express it as itself, like aside from its own DNA? Or?
Yeah, it's aside from its own DNA.
Okay.
So all these plasmids have promoters and they work independently, right? So they still have to enter the nucleus of the cell to be able to be expressed, but they don't have to work with the genome. There's a caveat there though, right? So, and I show you the example where we make endothelial cells, and I told you that only fibroblasts can convert into endothelial cells. Because sometimes it's not just about expressing something, right? It also has to function. So, and again, when we work with transcription factors, even if you successfully overexpress transcription factors in the cell, they still have to bind to the chromatin, to the regions of the DNA where they are supposed to actually drive function. If those regions are hidden, right, because the chromatin is packed, if they are not accessible to those transcription factors, then no matter how much you upregulate something, it will do nothing in those cells. So we think part of the reason why keratinocytes don't respond to the EFF cocktail is because the areas where they are supposed to bind are hidden, right? So even if we overexpress those free transcription factors, they never bind to the promoter regions in the DNA, and conversions never happen, right? So you have to, it's a balance between overexpressing too high enough levels and also making sure that the transcription factor is functional once it's translated into protein.
This kind of gives me, maybe you touched on something that kind of ties to what Danny asked as well, I was wondering about. So I know there's a lot of interest in developmental biology of like almost memory of how a cell becomes what it is, right? And the thing, what lineage it takes might change its epigenetics in a way, but it does allow certain things to be excess versus not. Even if it's like a functionally a muscle cell at the end, it can behave differently whether it came from a neural crest or second heart failure lineage or something like that.
Sure.
Have you looked into that at all, or is it your thinking?
So the way we have approached that is we can intentionally manipulate the chromatin structure, right? So when we want to convert a cell that's not easy to convert because of those regions just being hidden, you can treat the cell with acetylases to essentially make the chromatin more exposed. You can overexpress other transcription factors that induce chromatin remodeling, so it exposes the chromatin again. So yes, I mean, one approach would be to maybe pick cell types that differentiate or originate from different layers in the developmental stages, but you can also intentionally manipulate the structure of the chromatin to succeed at reprogramming.
A question, it was fascinating to see that model of Alzheimer's with quite impressive behavioral changes and histology as well. But when you showed the increased neuron expression in the cortex, so there was a lot of neurons that don't express neuron, and usually they're used with missile staining, for example. So have you looked into other structural changes, and do you think also whether the TMT had any implications on excitatory inhibitory balance? Because with AD, over time there are profound changes that result in seizures and epilepsy development. So behaviorally, it's really impressive components of the treatment. But what about the other outcomes? Have you looked into those, or were there any signs that it might be affected?
That's a really good question. So to your first point, the new one is indeed a mature neuronal marker, right? So there could be neurons present in there that are not expressing new N. And that's definitely something that we are aware of and we're looking into further, right? Just to see if there's maybe other neurons present there that might be contributing to different aspects of the phenotype that we observe. To your other question, it's also a fair point. We haven't looked beyond, this was about a year-old mice that we treated, but it's possible that maybe downstream we have other side effects of this therapy, right? Or things that we cannot mitigate, right? So far I only show you that we can improve spatial memory, right? But that's not the only component that gets impacted with AD, right? So there's still more studies to be conducted, and that's what we are working on right now.
So during, when it comes to like reprogramming, is the effects like immediate, or is it sort of like a delay in these functions? And so is there, during that delay cause like time, is there any sort of like side effects that lead somewhere else?
Can you repeat the question? I missed the first part.
So during reprogramming, is the effect of the conversion instant, or is there sort of like a delay period? And does that delay period lead to any like side effects?
So it varies from model to model, right? So we've seen that in some cases, a couple of days is enough for you to see the conversion, or at least major components of the conversion, right? Neurons is one case, and neurofilaments is one case. We've also played a lot with insulin-producing cells, and those usually tend to take longer to convert into beta cells, right? So, so yeah, it varies. The dynamics of the process varies depending on the type of model that you choose and also the type of cell that you start with.
All right, let's thank Dr. Gallego-Perez. See you all back here next week for Heart Week lectures.
I've been